All right, we're gonna start this next part. Um, we are gonna have a presentation right now by Katerina Nastou from the Novo Nordisk Foundation Center for Protein Research at the University of Copenhagen. So,
Thank you, Cecilia. So today, Martin has given a very nice introduction for me. I'm going to talk about S1000, which is a better corpus for evaluating species named entity recognition method. So let's start on why. Why do we actually need to be good at the identification of taxonomic names in text? There are several reasons, and people can argue what is more important to them. For me, we are behind the string database, so the most important reason is to do better protein-protein relation extraction, because doing good species named entity recognition can help us identify the source organism of interacting proteins when we are doing normalization of these entities. And this is an extremely important step if you actually want to extract what the text says and about which organism it talks about. But then, depending on the field, it is important also to do organism and any biomedical entity relation extraction. Extracting knowledge on the species and organisms themselves can also be very important. And in this era where we get so much data from microbiome, actually doing better microorganism identification is really, really important. So in order to answer all of these questions, we need an up-to-date corpus to serve the current needs of scientific research. But I said in the beginning, we are going for a better corpus. So what is already out there and why is this better? So the first corpus that was introduced, the first big effort, was L100, which was a corpus coming together with the Linnaeus tagger. And in that corpus, there were 100 full text articles with high quality annotation of all the species and organisms in this text. But the problem when you are limiting yourself with 100 full text articles can be that the diversity of organisms in this is low, and there is a high degree of repetition in the text. So this, when you train your models on top of it, can lead to overfitting, and you're overestimating your accuracy in comparison to what happens in the real world. In order to address this issue, a few years ago, Lars and other people in the group back then introduced the S800 corpus, which was, again, together with another tagger, the species tagger, and it's actually the same tagger that Ismail talked about that we use to tag everything right now. For that one, there was a different process in abstract selection. There were 100 abstracts from 8 different categories of text. Then these correspond to different categories of journals. So there were for protistology, entomology, virology, bacteriology, zoology, mycology, botany, and these were all the positive. And there were also 100 abstracts from medicine, where we really don't expect to find a huge diversity of organisms in the text or even anything mentioned at all. If people are doing medicine, they assume it's about human. They will not necessarily write that down. This corpus managed to become more diverse than L100. There were more than 3 times as many unique species names in that corpus, and it has been used extensively. So it has been used in other resources, and many of them come from our own lab. But what it has been lately used for is benchmarking NER methods. So because with the advent of the deep learning, large language models, there is, and also the ones that are specific to the biomedical domain, there is a huge need to benchmark those. I mean, it didn't start with those, it has started before, but now when we track the citations, we see that it's really going up and people keep using it. And I'm taking a paper from 2019, BioBird. I think a lot of people in this room will know about this model. And let's go to the table when we have the results on the biomedical named entity recognition task. The table is big. I will focus just on the first column. And you see here the state of the art on named entity recognition. And I think you can clearly see that the metrics are really, really lower for S800 than any of the other tasks in named entity recognition. And when we were looking in this, it really didn't make sense. It should not be that difficult of a problem to recognize species names in text. Why is this happening for deep learning models? So we started looking into what has happened in S800, why is it like that? And it was a good thing that I was in the lab that first developed it, so we could really go back and forth and discuss of what are the rules here, what is going on. So as with everything, S800 was a manually annotated corpus, so it all comes down to the annotation guidelines. And when we started looking at the guidelines and what was going on in the actual annotations, we started detecting things like some annotation boundary issues. And the reason for that was that correct annotation boundaries was neither a requirement in the original corpus, not considered when reporting F-score, because it was using flexible boundary matching of species names. So that is really not a problem when you're, for example, using a dictionary-based method. It will really capture the name, even if the boundaries are not correct. But that can really introduce problems when you're moving into deep learning, when the boundaries are inconsistent and the model can really not understand the context around it. Then there are other issues which we mainly attribute to how the annotation was done. Martin showed before the BRAT annotation tool, for example. It was not used in that case. It was more hands-on and not with a platform. So there were some fairly obvious species names that lacked annotation. There were some inconsistencies in the tagging of strains because the rule set was not that specific for that, and also in the tagging of genera. And then there were also some inconsistencies around the tagging of upper level taxonomical terms. For example, virus versus viral. Virus would be annotated, viral might not be annotated. So this really created problems. And yeah, this comes with the adjectival forms where they are not annotated, while the annotation guidelines said that this should be annotated. And then the one big problem and one thing that really throws the deep learning models off is the inconsistent treatment of common species names. The context around common species names and linear names is really not that different. So when you tag half of them and you don't tag the other half, the model is really thrown off. So we decided, why not fix this? It seemed like a good idea at the time. It took a bit more time than we initially thought it would take. So that's why we started doing the manual revision and the extension of the S800 corpus. And the revision and extension had 5 steps. The first one was the decoupling of recognition from normalization, and this step was called decoupling in later figures. And the one thing that was done back then is that annotations were added, and annotators were asked to add annotations even if they could not normalize them back to NCBI taxonomy, but some annotators didn't do that. So we made sure to stress that it doesn't matter if this can be normalized to NCBI taxonomy, we will annotate it anyway. And then there were annotations for names above species level. This served the tagger needs at the time. For example, when people talk about Arabidopsis in text, they usually talk about the species, while in reality, it's the genus, not the species. So we decided to go with the specificity in taxonomy, not with how people talk about these things. And then we did the revision of annotations for boundary consistency. So we put down guidelines to make sure that all annotators use the correct boundaries for the words, and we are not like using flexible boundaries. We say like, this is how a species looks, this is how far you should annotate. And then we run into the issue, what do we do with strains? Because in the previous implementation, some annotators had the strain mentions inside the species names, others didn't. So we decided that the cleanest solution for us at least was to separate the strain mentions from the species mentions and have two mention types in the text. So we introduced a separate named entity type, and the strain names were excluded from the spans. The fourth step was the annotation of genera. Because we started annotated strains, it's close to species, we thought, let's at least try to annotate the first level of taxonomy that is also closer to species, because especially in microbiome studies, this is also very important and very relevant to them. So let's at least try to do that in our text. And finally, because the original text was more diverse than L100, but we thought that we can still increase the diversity a bit and add more text into it, we extended the corpus with 200 additional documents and did a final polish to make sure that all the annotations are consistent and correct. So with the extension of the corpus, yes, as I said, it was to introduce the diversity, and we extended the documents, and that's where we go from S800 to S1000. So we decided to keep the original document partition of these classes and to avoid overfitting to specific journals or publication years. This is what was done in the original work. So it was just scoped within specific journals of these classes that I mentioned and specific publication years. So we decided to just ignore that, use a different strategy, and avoid overfitting to those as well. So the result for this, first of all, I will just show the corpus statistics for unique names and total mentions, what we did when we moved from S800 to S1000. And I mean, you can see the differences in all of the classes in the corpus, but I would like to focus basically that we have almost doubled with this reannotation effort and the extension, both the number of unique names, but also the number of total mentions in the text. And the nice thing is that we have a very high inter-annotator agreement in the task. So the rules are a lot, but we managed to follow them consistently. And this resulted in something very good. And we are really happy with the quality of the corpus right now. But we started with another research question. So this is used to benchmark NER methods. Has this revision and extension made the data adequate to train and evaluate a deep learning-based tagger? But at the same time, we didn't want to lose the initial focus. So this was a corpus developed to evaluate dictionary-based taggers. Can it still do that as well? So in order to test the first thing, the deep learning-based taggers, we did the experimental setting based on the methodology proposed by our collaborators in this work, Yoni and Sampo. And we trained the models, fine-tuned to detect entity types in the training data. So the three classes that we have annotated, species, strain, and genus. And we did multi-class classification. So we don't allow for overlapping matches in the entities. We split the corpus in the train dev test, again, following the original split that is introduced and has been used in all these years. And we did 8-fold stratified cross-validation. We did that because originally we had split 60-20-20, the test, the dev, and then we were getting these results and we were seeing we were doing error analysis on the dev set, and we were like, maybe that's not the best thing. It's a small corpus. Let's try to do a stratified cross-validation, and it paid off. It really, the different folds of the corpus had different things in there, and it showed up on the results. And then we compared the performance of 3 transformer-based models on S1000 based on the results of the latest BioNLP SER task. We decided to take 3 models that were performing really well in named entity recognition and evaluate the best performing model in the end on the different annotation steps to see actually how are we performing every time we are changing our annotations. So the first step, the evaluation on the 3 different transformer-based models, we used the Roberta BioLM, which is a specific Roberta large model. BioMegatron model, you can see which specific models we used below, and the BioBERT large model. They are, I hope you can all see, they're really all performing much, much better than the 75% I showed before, and all around 90%, but Roberta seems, as in many other tasks, to perform much better. And yes, it seems that species and genus also are easier classes. And I have to say, they also have higher support, more examples. Strain is more difficult, but it's also, there can be strain names that are just a simple letter O. So I can understand why models can still get confused with that. And the next thing is that we chose this best model and we did a progression. We tested the performance of species mentions in this case on the test data for the different corpus steps, and the arrows in this curve denote the sequence of the progression, and you can see in parentheses next to each step the F-score. So we start from a 74.8, what we see in the literature for S800, and you can see that even like decoupling, so adding all the annotations, checking for boundary consistency, and again annotating species and strains separately, and genera keeps us in the same F-score contour. And also what we saw is that introducing more diversity with the extra 200 documents dropped the score slightly, which was expected, but it was still way above 90%. And I have to say this number is a bit different than the previous one because it's just the species here. It's not all the classes. And then we decided to also evaluate on the entire corpus and test, as I said before, if we can do it both with dictionary and deep learning-based methods. And here we use the overlapping matching criterion, which makes the work of the transformer-based tagger even more easy. So we reach an F-score of 97%. And we did that to compare with the dictionary-based tagger to at least follow a bit how that method also works. And the score here was 84.7%. Of course, I should mention here, and it will be evident in all the results below, that the dictionary-based tagger is doing recognition and normalization at the same time. So this really can drag the results down. If we were trying to normalize the results of the transformer-based tagger here, it should also drag this number down a bit. And then we did an analysis to see the error categories that were coming up. What was the problem for these methods? And we can see that more than 50% of the errors produced by the dictionary tagger are called dictionary errors. And these are errors of things that are missing from NCBI taxonomy, common names that are not there, even linear names that don't exist. So these things can actually, like the dictionary-based method starts no chance on finding them. And then we have other things that are interesting that also show up only for the dictionary-based method, and this also makes sense. The discontinuous entities, we are not detecting those. And yeah, also unofficial abbreviations. Of course, the deep learning-based method can find them, the dictionary-based method cannot. And yeah, both methods have issues with ambiguous names. It seems to be also the main problem for the transformer-based model. So things that like bees, which can be both family and species, so they cannot detect them. And then, since the corpus allows us to do that, we decided to do an evaluation on a per journal category. So like this bacteriology and all these categories. And here with the overlapping matching criterion, we can really see that the triangles, the transformer-based method really goes just in the corner. It's crazy how good it can be in this task. And we can really see that it can detect it, all the entities. And then we see that, okay, yes, the transformer-based, the tagger really suffers in virology, but also this seems to be a more difficult class in general. This is the one that the transformer-based method is really has problem on detecting, and maybe more efforts and focus should go on this. And I have to say, this was the most difficult class when annotating. We really suffered with viruses. So in conclusion, we propose the use of S1000 as a gold standard for the evaluation of deep learning-based language models in the place of S800. So people who were using that, we suggest to use the new corpus because it's better fitted towards their needs. We have shown a clear improvement in training. The NER models, we had more than 18% increase in F-score. The expansion can allow training of generalizable models. We can still evaluate dictionary-based methods, and the corpus has almost doubled unique and total mentions. And all data, of course, are publicly available. We have a publication and all the data are available through Zenodo and GitHub, the code. And I would like to thank both my group, and I have bolded everyone from the group, like half the group is here at ISMB in different causes. And I would like, of course, to thank people at Turku NLP who have helped with training the deep learning methods and doing all of these evaluations. I would like to thank Tomoko for her help in the annotation of the corpus, and Vagelis with all his help in the error analysis and all the steps, and thank all of you for your attention.